Howdy everyone, you're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a Ph.D. program while trying to stay sane. We also often invite faculty from our interdisciplinary program here at Texas A&M and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M. In this episode, we'll talk to Dr. Seth Murray. Dr. Murray is a professor in the Department of Soil and Crop Sciences and Eugene Butler Endowed Chair in Agricultural Biotechnology at Texas A&M University. He received his BS in Crop and Soil Science at Michigan State University and Ph.D. in Plant Breeding and Genetics at Cornell University. His research focuses on the breeding, quality, and genetics of maize, aka corn. My name is Sarah, and I'll be one of your hosts today.
And my name is Veronika, and I'll be your other host today.
So hi, Dr. Murray. Thank you for joining us here today. We appreciate it.
Howdy. Thanks for having me.
Okay, well, can you please tell us a bit about your current research with maize?
My current research, I hope to expand beyond maize, but my current research and all my research has always been dictated by my graduate students. So their passion, their interest really drives the direction of the program. And then I sort of try and help guide that for things I think are fundable or things I think are relevant to the breeding program. So I was hired to actually address corn breeding, right? And so if you think about, there's 53 or so land grants plus HBCU land grants and tribal college land grants. And most of them had a corn breeder up until about 40 years ago. But with industry consolidation and the lack of a need for public sector breeding in the Midwest especially, most of those positions have been sort of transformed into corn geneticist positions or have sort of disappeared completely. So I'm one of the last 3 or so public sector corn breeders trying to develop corn varieties for farmers to grow the resulting varieties. Mm-hmm. And so most of my research really follows with this idea of how are we going to better genetically improve the crop. That's what my students mostly work on. And then we make crosses and do stuff that's probably not all that intellectually stimulating. Pollinating corn is just hot, sweaty, dirty, harvesting corn, same way. So a lot of it's just managing all these genetic materials, making crosses, and then screening the resulting material. I sort of avoided your question though, which is what's my current research? And the current research and the stuff I'm so excited about is saturating the phenome using maize as a case study or corn as a case study. And the idea there is that if we think back 30 years ago, people were using like isozymes and other type of genetic markers that nobody talks about anymore because SNPs have become so cheap and plentiful. And that's opened up a whole range of biological phenomenon we can discover, new methods for breeding using genomic selection. And so now with drones or UAVs, whatever you want to call them, we can start to capture phenomic features, and we treat those like genomic markers. So I think the most we have right now is about 1,050 phenomic features. And so we can use that to actually predict the performance of an individual better than the 150,000 genomic markers that the same individual might have, right?
So when you said that you're focusing on the phenome, that would be like looking at the different phenotypic varieties that your research is doing.
No, well, so we screen the phenomic features just like you would screen SNPs or molecular markers across a segregating population of individuals. Mm-hmm. We're screening phenomic features. So the way I like to describe it is if we think about, if either of you or the audience has a sibling, right? You and your sibling look similar. It's even better if you think about a twin, makes it even simpler, right? But if you think about your sibling, you look similar. Right? And most of that's due to genetics. But if we took one of you and put you in a war zone, you'd be 10 IQ points dumber, right? You'd be a foot shorter, right? Because it's not just your genetics, it's not just the nature part, it's nature plus nurture and the interaction between the two. And so when we're just doing genomics, we're only capturing the nature part. So what we find is if we can capture the nurture part with these phenomic markers, we can really predict performance a lot better. So if you think about you and your twin, or your sibling, if you're athletic, they tend to be athletic. If you're smart, they tend to be smart, right? So things are associated. And we can see the resemblance between you and your sibling, But if I was to ask a stranger to describe the resemblance, it would be totally non-informative. Like, you could not pick, based on that description, you couldn't pick somebody out in a crowd, right? You'd be like, oh, they're tall, they have brown hair, they have, you know, a big nose, right? That's not really informative. But those are the phenotypic features we normally think about. So what we're trying to capture is the phenomic features. So it might be like the distance between your nostrils, right? Like something a stranger would not normally describe. So that's sort of like the equivalent of a phenomic marker. It's not a phenotypic feature that we normally think of. So when people ask, what are we measuring? Well, we're really trying to measure anything we can that we can measure across segregating populations.
Okay.
Can you give us some examples of those phenotypic
Well, so
markers?
in Maize, in our Maize work, the main one that we're using is vegetation indices, just because they're the cheapest and easiest, right? Like, so start with the cheapest and easiest type of features you can measure. So vegetation indices are, if you've ever grown a plant, you know you can see that plant and you go, ooh, that doesn't look healthy, right? It doesn't exactly tell you why it doesn't look healthy, but you know, if you have some more knowledge, maybe you'd say, oh, it needs some nitrogen or it needs some water or whatever. But that color, that appearance of color is something that we can sort of see. And so the vegetation indices are automated methods of looking at ratios of color. So a camera typically measures red, green, blue. So you can take different ratios or different equations to calculate the difference there, and that's a vegetation indice. And the most common and most sort of useful one in plants and agronomy is NDVI, or Normalized Vegetation Difference Index, and that includes a near-infrared band of light, not just RGB, and that really predicts whether the plant needs, you know, more nitrogen or is healthy. But so we can take dozens or hundreds of those vegetation indices throughout growth, and that populates most of the phenomic features we're currently measuring, but we hope to expand beyond that.
And so these, the way that you measure these phenomic features, because I'm assuming you have a lot of plants, is this partly where the drones come in?
Yeah, so we, most years we've grown about 17,000 plots. So that'll be, you know, somewhere in the neighborhood of 7,000 genotypes and various types of replicates, maybe different treatments with irrigation, you know, nitrogen, whatnot. And so then we'll screen with drones throughout time, so ideally multiple times per week, and get these vegetation indices. We'll also get things like plant height, and then we've worked on a number of equations to try and come up with other phenomic features we can extract from that. But that's, I think, the new frontier, is how are we going to expand that phenomic feature list to be, you know, 100,000 phenomic features to complement the 100,000 genomic markers.
So all the plots that your drones are overseeing, are they in College Station only, or
so that
other
was
places?
You know, we need to think about where to put resources when we do science. And when I mainly focused on breeding and molecular quantitative genetics like QTL mapping and GWAS, we used to do trials across the state, a lot of trials across the state. We'd have like 7 to 10 locations. And you really need that if you're breeding a crop, because you want to make sure it works in every environment, right? But I think over the last 5 years, I've sort of realized that, you know, I can make a much bigger impact in science than I can in breeding a new variety. I mean, both those things, there's a lot of potential for impact for, but the science, you know, nobody else is really doing this phenomic work right now. And so we reduced the number of locations to just College Station. And the way we get different environments is we'll plant it throughout the growing season, so at different dates. So those plants, as they grow, are exposed to different environmental conditions, heat, drought, whatever. We'll also irrigate or not irrigate, or apply more nitrogen or no nitrogen. So those are the ways we get different environments, is by management.
Okay. Do you find it difficult to grow the plants here in College Station during the summers when it's 90, 100 degrees a lot of the days?
So the average yield of corn in the United States is like 185 bushels an acre, which is, you know, a bushel is 56 pounds. In Texas, our average yield in Central and South Texas, where my research program focuses, our average is about 85 to 90 bushels, so less than half the U.S. average.
Oh, wow.
Whereas in the high plains of Texas, it's 200 bushels per acre. Now, the high plains experiences just as much heat, but what they don't have is the high nighttime temperatures we have. That's really the big stunter for our corn, is just, you know, corn likes a cool night, and we just don't get that.
No, we don't.
But the biggest things that keep ruining our experiments over the last few years isn't just the drought, it's the weeds. So being able to control the weed pressure is really difficult here.
Do you use pesticides, or do you try to go with natural weed repellents?
Yeah, we use herbicides. No, trying to do anything organically in Texas is extremely challenging. Up in the Midwest or Michigan where I'm from, it's not, the weed pressure just isn't as great. But here, the weed pressure is so great, we put a pre-emergent herbicide down, which prevents the weeds from starting. And then throughout the season, we'll spray. But our varieties, our research varieties, are not Roundup ready, so we can't spray over the top with glyphosate like a farmer could. So that's another big challenge. And then this year, anytime we could spray, we got so much rain that the sprayer couldn't actually get into the field. So hopefully by next year, drone spraying will become a more common thing. But not from our program. We'll work with the weed scientist and the farm to spray our crops with drones.
Mm-hmm. I'm curious, what are the current problems facing maize farmers?
I think the biggest one, if you look at the history of agriculture, we've gone from 40% of the population was farming maybe a little over 100 years ago to less than 1% of the population of farms now. And you can see that as a victory because that's allowed all those other people to specialize into things that are not so difficult and laborious. And it's been done through technology, right? So bigger tractors, better varieties, bigger combines to harvest everything. That's been a huge improvement. But as that's happened, the value of corn, in real dollars, continues to drop. And so farmers have had to grow their operation. So now farmers in Texas, I don't know the exact average farm size, but let's say it's 1,000 acres. So there's people who are 10,000 acres, right? And so they're dealing with these really small margins because by the time you buy the seed, you put fertilizer down, you irrigate, you get trucks to move your corn. A lot of farmers sometimes are in the red. So it's usually the federal subsidies that help, make that into a, enterprise that's actually in the black. So I think it's the low price of corn, and we see, Brazil has come online with a ton of corn. China is rapidly increasing their production. Throughout the world, corn yields continue to go up. It's a major success story, right? Corn is so cheap now that we pile it up outside in piles in Iowa because they don't have enough place to put it, right? But that's in a good year. So the reason that we still need to be concerned is not every year is going to be a good year, especially with climate change. Mm-hmm. And if Brazil experienced a drought and we experience a drought and China experiences a drought, then we may actually have a shortage of corn. So there's a reason we've had this plan, like a federal policy, to make corn as inexpensive as possible because you can't always account for the bad years, right? And then you have a good bumper crop and you don't know what to do with it and no farmers make money on it. So this is always sort of the challenge, is that we want food to be cheap and accessible. We want feed to be cheap and accessible. We want to have ethanol to reduce our climate emissions. But it's really hard for a farmer to make money when the goal is to produce more and more and more corn.
Do you think some of the research that you're doing with the drones and looking at these different markers, do you think that'll eventually be able to be applied to farms so that they can increase the yield that they get and make the good years really good?
Yeah, so one of the things we've discovered is, for instance, that there's some time points that, at least in Texas, are really important for the corn growth that physiologists, sort of overlook because they were really hard to measure, especially early in growth. It's really difficult to measure those corn plants. So it may deal a little bit with farmer management, but ultimately we're developing our tools to select the best varieties,
okay,
right? And not just for the application of developing better corn, but also, from this phenomic approach, I see real usefulness in humans, other animals. A lot of that's possible. And if we look at the history of science, both genetics and statistics mainly came out of plant breeding and plant sciences. And so I think this phenomic approach could be the same thing because we don't have ethics concerns. I can create as many babies as I want from my corn plants, and I can kill them all, and nobody cares, right? But when we move into other species, if you did it in cows or pigs, there's a lot of cost just to raising a cow or a pig. There's virtually no cost per corn plant. So we can use that as a laboratory, and unlike doing something in a greenhouse or growth chamber, we virtually have unlimited space. So we can get up to 17,000 plots, right? So the statistical power you can use to find things is really expanded by having a field program.
I didn't think about that, but yeah, the statistics would be more sound the more plots that you have. So that's smart. Did your work with the phenomic markers, is that part of what led you to found the Plant Phenome Journal that you did?
Yeah, I was just, so my primary scientific society, so I was just the president of the Crop Science Society of America. We have two sister societies, agronomy and soils. So altogether it's about 10,000 members, but crops is about 4,000. We were way behind the boat on genomics. So the Plant Phenome Journal is our highest impact journal within the three societies. And that was founded well after a lot of the competing journals. And so I said to my society back in 2016, I think there's going to be a lot of opportunity on the phenomic side, what we're seeing in drones. And so I had to get board approval and all that. But we founded that journal because I thought, plant science can really lead this. So for too long now, you look at Pritchard's omnigenomic model, right? And so that's, lots of small effects and context dependency and all this stuff that plant scientists knew for a long time, but plant scientists don't necessarily get the recognition for that, right? You have to do it in humans for it to be exciting. And so I really want to position my field and my domain to be leaders again and not continue to have atrophy of positions throughout universities.
So what would you say that your biggest finding is from your lab so far?
Biggest finding from my lab?
Or something that you're like the most excited about?
Well, those are two different things. The thing I'm most excited about, I think, is, that this phenomic approach is working. But I think the biggest finding in our lab so far, our major study was, I guess they would be the same thing in that case. But what we haven't talked about is the way I got into
right.
plant breeding. Mm-hmm. So in high school, my teachers gave me a book, one of my English teachers, called Becoming Native to This Place. And it was about how most natural systems are perennial polycultures. So if you go out into a forest or you go out into a park, you don't see an annual monoculture, right? You don't see just one species, and if you do, it's a perennial species, like a birch forest or something like that maybe. And so we look at our agricultural system and we're really working against nature. And so I love this idea that we can breed perennial polyculture systems so that we'd have multiple species and that we wouldn't have to tear up the soil every year, which leads to a lot of greenhouse gases, leads to a lot of erosion, a lot of burning of fossil fuels to plow it up every year. And so that's really my career goal. That's really what I want to work on, but it turns out there's really no jobs in that. Not only that, you can't get any funding for that because it's too disruptive to our current system. So it's really a career type project. I think my biggest finding for me personally on this so far was perennial maize. Most people have made the cross, or have always made the cross, using the perennial parent as the male and the domesticated parent as the female, right? Which makes sense. Corn's got these big ears. You cross the male pollen to it, you get a lot of seed from that cross. It appears that from my work, and we've only sort of published this in sort of low-level things, that actually there's probably a cytogenic effect. And so you need that maternal cytoplasm or mitochondrial genome to actually get the plants to be perennial. So making that cross in the other direction seems transformative for delivering, at least in maize, a perennial plant. So nobody really cares about it, so that's why I wouldn't say it's my biggest finding, but for me, I think it's the most impactful. It changed the direction of the way we do perennial breeding.
No, I think that's interesting, though, that a lot of science can get stalled sometimes because it's just we do things in the acceptable way, the way that everybody's been doing it. And then occasionally, if you just come along and switch it up, you can find some really interesting things from that. So I think that's actually a really interesting finding.
So that's been transformative science, and then technology is the other thing, right? So there's lots of things people in the past couldn't do, but now, because genome sequencing is so inexpensive, all of a sudden you can do these experiments that you never could do before. Or now because drones are cheap and ubiquitous, we can do these things that may have been logical, but nobody had the tools to do them before.
Overall, what would you like your broad impact to be? Just all of your research for the rest of your life. What would you like to leave as a legacy, essentially?
Yeah, I mean, the major legacy is the students I train, right? They're going to make way more impact. The chance that I'm going to have something that's going to change the world in my research is almost zero. If we think about our all ability to do science, I mean, you may have a little impact within the scientific community, but the chance that your students will end up having an impact is multiplicative. So the students are clearly my biggest legacy. I think I've had 35 that I've chaired or co-chaired now. And most of them have gone to industry, but one of them started a company in Texas, in South Texas, that's doing phenomenal, supporting seed companies throughout the world and trying to grow stuff in Texas. One of my current students is the breeder for corn nuts, if you've ever had corn nuts.
Really?
Yeah.
I've never heard of that.
Oh, that's cool.
And then I have many other students in various industry positions, teaching at small colleges. And then one of my students was the soybean germplasm curator for the United States. So if somebody needed, soybean germplasm from the germplasm bank, he was the person who was in charge of that, which is a very important position for food security throughout the world. So that's where the impact really can come from.
Mm-hmm. So what would a typical day for you look like in the lab?
Well, I don't have a lab.
Okay.
And I've never found this elusive typical day. The thing I love about my job is I can just sort of wake up and do whatever I want that I think needs doing.
That's great.
So for me, that's really great. For other people, I know that they want a little more structure in their lives. But being able to work on anything that piques my curiosity, or I think needs doing or could help somebody. I mean, that's another big thing is spend a lot of time, informally mentoring people, right, about job applications or navigating grad school or whatever. I think that's where a lot of impact could be. I would hope my legacy would be the start of perennial polyculture systems, but I just I'm 16 years into my professional career, and I still see very few inroads for people supporting that. If we think about how long it took to domesticate crops, it was thousands of years, and those were annual crops where they have very fast cycles. By definition, a perennial crop should take twice as long. But now we have all this cool technology, so maybe it can go faster. But it's a real, I see a problem that needs doing. That's what I try to focus on. But you need to have resources and you need to have some community buy-in to make that happen.
So you mentioned that you do not have a lab. So what do you have then?
So we do almost all of our work in the field.
Okay.
We've used the Texas A&M high throughput phenotyping greenhouse. And then we have an office space where, most of my students now are data science focused. Mm-hmm. So, we typically hire a bunch of undergraduates to help pollinate the corn and do a lot of that work. And then we'll have one or two field people, technician, grad student, who has a field-based project. And the rest now are mainly analyzing, data. So.
Mm-hmm. That makes more sense, yeah. I was just confused on when you said, I don't have a lab. So,
right.
But you have grad students and everybody that work with you. So
yeah, absolutely. Yeah, I think a lot of people, when you say lab, they think of like pipetting. Mm-hmm. And I did that in grad school. I'm hoping for better things for my grad students, right? Like, I feel like we shouldn't train people for jobs that have existed in the past, and robots and AI are taking over a lot of jobs. Mm-hmm. We should train students for things that are happening in the future. So like, I tell all of the students whose committees I'm on now, like, I will not approve your dissertation or thesis unless you use ChatGPT to help write it, right? Because that's the tool you should be learning.
That's a very, very interesting comment.
That is an interesting take. We do have, there's the side of professors that definitely agree AI should be used, obviously responsibly. Mm-hmm. But then there are others who think that maybe it dumbs down the process a little bit because you're not making the thought connections yourself, the computer's making it for you.
Sure.
And so it's refreshing to hear that. I don't think I've heard that.
Yeah, because me personally, I use it a lot, even just for like when I'm in the lab, like doing the pipetting, because that's what I do. But just like, I don't know, sometimes there's not people around me to like answer my questions, but I still have the questions and I need them to be answered for me to move on or continue my work, and the AI really helps.
Yeah. But I mean, the responsible and trustworthy, right? Like there's a pendulum that can swing from like, don't use it to use it for everything.
Of course.
And, you know, I hold people accountable for what they turn in. And if what they turn in is crap because they use ChatGPT to write it, then that's on them, right?
Yeah.
So it is a responsible use. But I think there's really good, you know, most students that I've seen that have had problems, all my students who have had problems with graduating, if it wasn't, you know, a family or mental health issue, which they have no control over, it's with writing. And so I think the best thing ChatGPT can help, you know, ChatGPT is like Kleenex. There's lots of other AIs out there, but that you can use a tool like ChatGPT for is helping you write, right? I think that's one of the primary goals, making an outline, right? Helping write, and then you go and rewrite the stuff that it says with your knowledge. I think the other way I heard you say that you sort of use it, Veronika, is the way that I used to use Wikipedia, right, for getting some background knowledge. And I remember when Wikipedia came out, you know, professors were very much like, don't use Wikipedia. That's not, you know, it's all junk.
Yeah, like you can't trust it. And
it turns out Wikipedia is actually pretty reliable, right? There's a good community of people who fix it. And then I think back to the time when I was in elementary school and I was told, you know, well, you can't use a calculator on this exam, right? You're not going to have a calculator in the future.
We do.
And right?
We do.
Like the jobs that you guys are going to go into, the expectation is that you're going to be as efficient as you can be with whatever tools you can be. And that's going to involve use of some AI. So you should begin to learn it now and be a leader in using it rather than playing catch up once you get into the job.
So like, still about AI, I had to do some coding. I've never done that in the past. I just took one class about it, and I did use like an online AI software to help me write a code for what I needed it to be used for. And it was very helpful because I can't imagine doing that myself with the knowledge I have. But then I took it to another member in the lab, and they were questioning it and asking me if it's actually considered plagiarism and stuff like that, which made me think, but I said that I do not think so because it helped me write the script, but the data is mine. And if I would need to, I would obviously like cite the source that I used to write the script. But it was just a strange conversation because they didn't really appreciate that it didn't come from my head, basically.
Well,
I mean,
you're not going to run into when it comes to coding, at least. There's a bunch of different ways to do coding, but just because two people choose the same way to use the code doesn't mean they're plagiarizing each other. So
yeah, lots of code is made as packages for other people to use.
Right.
Right. But we think about the opportunities now for people in the bioinformatics space, for instance, with this. I mean, I was really impressed with Aaron taking our code and translating it into Python code. He'd never learned Python, but with ChatGPT, basically doing that for him, right? And then you think of all these people who have trained to be coders in various things, and they're basically going to be displaced. So I think in some ways it's also a cautionary tale, right? That don't train yourself on a technology, because that technology is going to go away. It doesn't matter what the technology is. Train yourself on how to use technology and think about the problem at hand and how to solve it. And then you'll always be relevant in whatever you're going to do. So I think rather than take more classes in coding, I think, you know, in my opinion, you did the right thing. Use the best tool to get done the problem that you need to get done, you know, and then acknowledge it. Acknowledge, yes, I use ChatGPT. If, you know, what matters in science is the results. The process is not, you know, the process you can geek out about, you can discuss it, but really it's ultimately what did you find? And too often we focus on the process as, you know, your Ph.D. is a philosophy degree, and your results that you get from that are the things that are important. If I gave you a recipe, do this, do this, do this, do this, that certainly doesn't equal a Ph.D. in my book, right? But I think sometimes that's when, especially graduate students, when they come in, they think they need to learn this technique or this technology, but that's going to go away. If you just learn the recipe and you don't learn how to adopt new technologies and think about the problem and solve the problem, I'm not sure how useful your career is going to be, right, or impactful.
Oh, this was a great detour. Very refreshing.
So is this one of the things that you try and instill in your graduate students to, you know, learn how to use the technology and adapt with it?
I hope so. I mean, the thing that I've realized as I've gotten older is you can try and communicate a message, but that isn't necessarily the message people are listening to or really take from it. So I hope by encouraging people, like mandating you need to use ChatGPT or a similar technology to help write your thesis, I don't care if you end up using it in the final product, but the fact that you had to go through the process and learn it and go, oh, it's good for this, it's bad for this, I think that's the valuable part, right?
Are there any other particular skills that you try and instill in your graduate students? You said you'd had quite a lot of them.
My philosophy for graduate students is I try and find their limiting factor and help them work on that, usually through passive-aggressive, harassing comments. Probably the best example of that is I had a guy in my program who was just really kind of I don't know if I can say that on the podcast. He was really kind of
We can beep it out if we have to.
And so I worked with him for a long time about like, hey, this type of behavior is not going to help you in the future when you go to get a job. You'll be fired from a company by doing this. And I think that probably has led to more of his success than, teaching him how to use technology. Right.
Yeah. I think a lot of grad students get in their own way without realizing it. I know that I've done it plenty of times and will probably continue to do it until I graduate. And sometimes you just kind of need that smack upside the head to pull you back to reality and go, hey, you need to fix this, or you're just being a detriment to yourself at that point.
Yep. Yeah. Here's the funny thing that you won't realize about this until later. That feedback that you get from your advisor, from your committee, or from your teachers in classes, some people aren't really great at taking it, right? Or, most people aren't like, oh, thank you so much for the feedback. But then as you advance in your career, there comes a point where you just don't get critical feedback from people. You might get anonymous comments from reviewers, but like if you're an asshole at that point, nobody's gonna give you any feedback on that and be like, hey, they just won't work with you. Right? And so I think the helpful feedback that we give grad students, however we can do it, is really probably the most important thing for helping them advance in their career.
No, I agree. It's definitely without the feedback I've received from my committee members and my PI, I wouldn't be this far in the program. I probably would have dropped out and gone home by this point because, it's a PhD. It's not meant to be easy, and you spend a lot of the time feeling dumb, and it's hard to stop feeling dumb all the time without feedback and picking up on where you're doing things maybe a little bit wrong. So I think it's helpful.
Yeah, I agree.
Is there any advice that you would give to students that are wanting to get into plant genetics?
Any advice? I think the same advice I'd give to any student would be, and it's not always helpful, but you need to find a passion and have some direction you're going to. It's not really helpful saying, I want to get a PhD, right? You need to have some type of problem you want to solve to be involved in it. So I think the students who find plants interesting, it's great. But I don't try and recruit anybody to see the world as I do or come work on what I want to work on. I don't think that's really helpful. We're all most effective when we work on the things we find interesting. So finding whatever your passion is, and if you don't have one, arbitrarily determine one, because at least that gives you a guidepost. And then you can, as you look at getting pulled a million different directions, you can say, is this what I decided sort of my goal or passion was, and focus on that. But if you don't have any, I think I would discourage you from even attempting grad school, because I think you need some type of guidance to take the self-abuse and, academic abuse that you get, and, sort of tongue-in-cheek academic abuse, right? It's not easy. There's a lot of freedom, there's a lot of opportunity, but from a mental health standpoint, you don't have necessarily a clear direction unless you set it.
Okay, well, talking about mental health, what do you do in your free time to stay sane? I know you have a farm.
I do have a farm. What's
on your farm?
I have about 500 fruit trees, although A lot of my apples have died. I've planted about 250 apple varieties, 60 pomegranate varieties. I have a little over 200 pomegranate trees, peaches, plums, figs, blackberries. So like even this morning, before I came here, I was finishing filling up my water tanks for when I go this evening because we don't have irrigation set up everywhere. And so I spend a lot of time watering at this time of year. The rest of the times of the year, I spend a lot of time on my tractor shredding walking paths and, mulching fruit trees and fertilizing fruit trees. So that's a lot of my activities, or just fixing stuff around the farm. Tractor's always breaking.
Do you sell what you grow on your farm?
So I'm a plant breeder. So I like to call it my academic exit strategy, which is in none of these species has anything been bred in the South. For the last apple variety in the South, I think, was at Reverend Morgan in 1880 or somewhere around there. So I think there's a lot of opportunity for, whether it's home gardener or eventually commercial production, for crop species or perennial fruit and tree nut species. And there's really nobody addressing it. I mean, it's mainly hobbyists, and I guess I'm kind of a hobbyist. But I like to think that my experience in corn breeding will make me a little more effective at my experience in fruit tree breeding. But I couldn't get a job at any university or any company breeding apples for the South. There's just no perceived market, right? There's no perceived return on investment. But if you had a variety that, I think usually the licensing fee for a variety of, let's say, an apple tree is somewhere between $1 and $10. Right, so you think about 100,000 apple trees being planted of your cultivar, which would be sort of small, right? But that's actually turns into, something of a retirement income.
Yeah, I know up at least near where I'm at, they have the orchards that you can pay your $5 and go pick your berries and everything. And I know I always enjoy doing that. So I just didn't know if you offered something similar down here.
No, we've talked about it because this is the first year. I've only owned the farm for 7 years, and I've been slowly planting stuff. So this was the first year we got about 1,000 pomegranates. So in my undergraduate American agriculture class, I got all the arrows out of 5 different cultivars. We did a little taste test. It was the first time many of them had had pomegranates. Pomegranates do amazing here. I barely have to irrigate them. The grasshoppers don't eat them. The deer don't eat them. Apples, it's just they just keep dying, and they need so much water and so much care. But there's a few varieties that are really doing amazing. So, so far I have not really had to deal with a fruit glut. But I'm also a brewer. Okay. I've been brewing since before. You can't buy alcohol till you're 21, but you can buy the ingredients to brew beer much earlier. So I like to take the fruit juices, back when I lived in South College Station, we had a pomegranate tree, and so I press out the juice, and then, you could make a wine with that, but I like to mix it with the beer ingredients and co-ferment them, so it's not like a fruit beer where it's sweet, it's more like a cross between a beer and a wine. And I find my wife likes that a lot more than my beer.
That's so cool.
That's an outlet for some of this extra fruit.
Okay.
That's so cool.
Do you have any hobbies that are unrelated to plants that might surprise people?
No, just mainly welding and building stuff. I mean, chainsawing. Love clearing brush on my property and chainsawing and yeah.
Simple things. I like it.
Amazing.
Spending time with my wife, walking dogs, and clearing up the property and improving it.
I mean, that's always nice to hear.
Yes. So do you also have farm animals that are part of your farm?
My wife has chickens, but
Okay.
we started with 30 and then hatched some more. I think we're down to 6. So it's almost time to order some more. I mean, these are 4 to 5 year old chickens now, so it's amazing. They're still even laying eggs, but they're her girls. So
That's so cute.
That.
I would have some chickens if I could, I think.
yeah, they're really fun animals to watch, but they are absolutely filthy and disgusting.
Yeah, I stay on a ranch, and they have chickens, and they run around like crazy. And I love them. They're so big and fluffy, and the little waddles that they do across the road is hilarious. But I don't know if I would want to take care of them myself, but I also don't know if I want the extra work, especially in grad school.
It's a lot of work.
Yeah.
But yeah, watching him run and waddle back and forth, just like every time I burst out laughing.
Oh yeah, it's great.
Oh my God, this is funny. Kind of going back maybe to science, I have a question, if that's okay. So where do you think the field of plant breeding will go in the future?
Great question. Where do I think the field of plant breeding will go in the future? So, again, agriculture is probably more than any other field I can think of advanced with technology. So I really believe in this phenomic. I think this phenomic thing is going to make a big change. What we saw in the 90s was this belief in biotechnology or GMOs, right? That somehow we were going to edit a genome and we were going to breed crops through that way. That's been a colossal failure, right? There's Roundup Ready and there's BT and there's a couple other genes like Drought Guard even hasn't been that successful. So it's been a colossal failure maybe because of the science. First you have to discover a gene that actually works across genetic backgrounds, and there's some articles from industry that show that is an extremely rare event to find a gene that actually works across genetic backgrounds. Then you have to transform it. But then there's this other issue of you have to get acceptance by the community, right? By consumers, by policymakers, deregulation. So what we found was these companies, this major seed breeding companies, laid off a lot of their plant breeders thinking, well, we're going to do biotech for everything. And it's been extremely profitable when they've been successful, but it's kind of like a unicorn to have those big successes. So then they started hiring their plant breeders. Oh, no, we really need plant breeders back. Well, we're seeing a similar trend now that we're going to use AI and drones, and we're going to eliminate all our field-based breeders, right? And I think that's also going to fail, because ultimately you need people who are sort of responsible and have that passion for, like, I like that line or I like that variety. I'm going to fight for it to succeed and get to the farmer. Because you can do all the tests in your experimental farm, you can scale it up, but until a farmer actually grows it and on large acreage over multiple years, you won't necessarily see the problems with it. So that's sort of what happens. We start with this sort of inverted pyramid of like, oh, all these varieties look great, and then you start planting them in more locations, more people grow them, more environments happen. Oh, that one's crap. Oh, that one's crap too. That one's crap too. And then you get to one or two at the end of the pipeline, and then the farmer grows them, and maybe they say, well, that's just crap, right? We don't like it because it's ugly, right? I've heard that before from a farmer. That's an ugly lie. I don't like growing that hybrid. It's ugly, right? It doesn't have any impact on their end results, but we're all sort of emotional creatures and aesthetic creatures. We don't like the look of it. It's fine. So I think ultimately, the saddest part about plant breeding is there's fewer and fewer people involved in agriculture. They're larger and larger organizations. And so now, like, a farmer's making a decision on 10,000 acres, right? Instead of 100 farmers making a decision on 100 acres, as it used to be. And so what we're going to see is more and more concentration and less people, and a lot of it, more of it going to industry, except for really specialty crops. Did that answer your question?
Yes, it does. Thank you.
Other than that, is there any words of wisdom you would like to tell anybody who would potentially want to apply to the genetics program here at A&M?
Yeah, that's a great question. I think I'm really impressed with the direction of the genetics program over the last 5 or 10 years. I think, I like how you guys have a cohort.
It's nice.
I like the rotations, even though when I was a grad student, I declined the rotation and just chose to work with an advisor so I could, find my project and get done sooner. I think the rotations do have a lot of value, especially your 1-month rotations. I don't love the rigidity of some of the classes and the seminars don't necessarily fit a lot of times with my students, right? Like molecular protein interactions and plant breeding are about as diametrically opposed as you can be. So I struggle a little bit with that, but I think that the genetics program is so, the cohorts are so strong and set up for building successful students in the future, right? That I really do push a lot of people into going into it. And I think A&M's got one of the better genetics programs that I can think of now. I also think that, a huge advantage we have, and it's not throughout the whole genetics program, but at least most of the people I associate with in genetics and like my own department, everybody gets along for the most part. It's a communal, supportive environment. That's part of what's kept me here at Texas A&M. I know plenty of departments where people fight, they deny each other tenure just because of stupid reasons, not because of success. And I think it's just a really good environment, and I want to continue to help maintain that environment however I can. I'm not sure if that touches on some of the...
That was great.
Okay.
No, I think that was great. I mean, I think the same thing. I love this program. So,
Me too.
Yeah. But it's not for everybody. I mean, there's no one program for everybody, right? So my mentorship style is, and my lab leadership style is it's not a lab.
It's not a lab.
My lab leadership style is I let the inmates run the asylum because, again, the grad students should dictate the direction of the program. I shouldn't just be telling them what to do. That can be very difficult for people who like a lot more structure. Then there's other programs where, again, the advisor will sit with them every day, tell them you need to do this next, you need to do this next, you need to do this next. I think that's great for some students. I just don't think it's going to develop great people to do science after they leave the program. So I don't have lab meetings. I don't even schedule meetings with students. I just have an open door, and when I'm in the office, they just stop by all day, and they can always schedule a meeting with me if you choose to, but really I think that helps develop the independence, I believe. Maybe it's my confirmation bias, but I think that helps develop the independence to have people leave the genetics program or the plant breeding program and go be leaders, right? Which is really what I want to see. I want to see our students be the leaders in the country of whatever they go work in. Don't just do it half-ass, like absolutely dominate whatever you're doing and have a passion for it.
Yeah, I do think one of the important parts when choosing what work you're going to do is finding an advisor that has an advising style that works for you, because each student requires vastly different things. And so some people, might need more structure. Some people might really crave the freedom of it. And so I try to tell the newer students that are coming in that that's something they should be thinking about when they're going on their rotations is the way that person advises you, does that make you feel confident in, proceeding and making progress for the rest of the time that you're here? Are there any other things that you can think of that genetic students might need to look for in potential advisors?
We met with the first year students, and I had a list of a number of things here, but I think number one is communication. First of all, I think your advice is great. The fit with the advisor is the most important part. I mean, in genetics, they don't get you in, but in my department, home department, that is how you get in, is with your advisor. But in any program, that's how you get out, right? So you have to be able to, can I work with this person for 5 years? But I think communication is number one. I think when I talk to the first year students, I put it as number 4 and 5 too. And communicating with your advisor is so important, right? I think most advisors, I'd like to think all, but I probably know better, understand if you're going through, some family issues, or if, your dog died, or, whatever happens to you, if you communicate that, I think they will respect that, want to help you and work with you. If you don't communicate that and it's just radio silence, then that really, I think, frustrates any supervisor. It doesn't matter if it's an advisor in graduate school or even if you're working somewhere. If you don't seem engaged, or you're not present, or you're not delivering what you said you'd deliver, if you communicate why that is, I think people want to help you and work with you and help you be successful. And if you don't communicate that, that's a real challenge. And different advisors have different communication styles. I've gotten some students I only text with, just about. Some students I only WhatsApp with. Some students I only email with. Some students I only see in person. Other advisors may have different, preferred communication means. But communication is definitely number one, and being able to communicate. And And being able to share your failures as well as your successes, I think that's really, really critical, because we're all going to have some failures, right? I believe you learn more from failure than you do from success, and that's a big part of grad school. You know, you want to learn stuff so you can get out and not have that when you're employed, and a failure is much more problematic. So people who are supportive of your failures, I guess those are a couple things that come to mind.
Yeah, I mean, everything that you mentioned helps to build the relationship with the advisor that you will continue even after you graduate, which is so key.
Yep. You know, I know a student by the name of Aaron DeSalvio who talks about building relationships as being one of the main things, and I think he has that right. He's about the only student I can think of that could work with a couple of my staff members or postdocs who the rest of the lab seem to have problems with. So he's all about building positive relationships with everybody, keeping a positive mental attitude.
Yeah, we love Aaron here at the podcast, just for the record. Yeah, I really like the students that are in our program. I mean, we may not see each other very much, but we're all very friendly, very supportive of each other, very happy when, you know, someone gets a publication or a grant or anything along those lines. It's just, you just kind of feel happy for them because they're doing something different than you, but you want them to succeed because we know they're good people.
Yeah. I think, and that culture is really important too. When I was at Cornell, one of the things I think was so important was we did have this very collegial, supportive, but also, you know, competitive in the positive way culture. Like, oh, that person published a paper. God, I really need to get started on my paper, right? And I think that's good for elevating everybody to, you know, not have like a negative competitive culture, but like, you know, maybe like a friendly volleyball match or something where, you know, you want to win, right? You want to have the most publications or whatever, and so you're going to work hard towards that goal. And I think that's a positive thing, and I think it makes everybody, you know, better. And I haven't seen the backstabbing competitive cultures that I hear about in some schools or some disciplines where it's like somebody would sabotage something, right? It's a positive thing where you are happy for them, they got their publication, but it also makes you like want to try a little harder.
It pushes you.
Yeah. I think a little bit too much of that can probably fall into a bad space just because of the interdisciplinary nature and the fact that we're all kind of coming in at different aspects. Like some of the cohorts will already have a master's, so of course they're going to graduate a little faster. They're probably already going to have some publications, and so I think there's a line to draw for comparison or competing, because sometimes there is no friendly competition there because it's a little bit outweighed. But I definitely agree that seeing other people that you have respect for succeed makes you want to put in the extra effort to also succeed as well.
Or when it comes to publications, if you work in bioinformatics, you can get stuff done a lot faster than somebody who works in yeast, and the person working in yeast can get it done a lot faster than somebody who works in field-based plant breeding, who can get it done a lot faster than somebody who works in trees. But publications, you know, I think we oftentimes put too much weight on publications. So I usually like to bring up examples that are not common. But I'd say service, right? Like your job running the podcast, right? Like if other students see that you're running the podcast, I hope they say, well, I should do something like service oriented, right? Or TAing, right? Like I want to get the best evaluations on my TA, or I want to have the best connection with those students, or have the most students that I TA go to graduate school afterwards. So it's not just publications. There's other ways, but you want to see, I think you want to be in a cohort of people who are all successful in different ways, and they push you by their successes to try and be better.
I agree.
Yeah, that was great.
Well, Dr. Murray, thank you for joining us today. We really appreciate you letting us pick your brain. Today's episode was hosted by Sarah Ingram and Veronika Mojic and produced by Anthony Matarazzo. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, and X at Genetics Podcast. Thank you for listening, and we hope you'll tune in next time.